Hi everyone, this is Christina Wallace. Welcome back to the first term of Human Anatomy and Physiology at Clackamas Community College. Today we are going to talk about the histology of nervous tissue, which in your book is exercise 15. So here we are at your lab report. I just want to start you out and note a couple pages. Your lab report, exercise 15, runs from page 251 to page 258 in your lab manual. And also, your lecture notes for this section will be very helpful. So your overview of the nervous system lecture notes, pages 4 to 9, are very helpful for this lab. So this lab is all about the histology of nervous tissue. So you're going to be looking at the cells that you find in nervous tissue. There's a couple types, and they have vastly different functions. And then we're going to be looking at 5 different slides of structures in the body that are made of nervous tissue. And so I'll go to all the links in the instructions. So here we are at your online instructions. And so I've kind of done a little bit of a nice thing. I've color coded all the slides for you. So our first slide is going to be the slide of the central canal. So this is slide 1. And so, and this is kind of goes along with your lab manual. There's pictures in your lecture notes that talk about this. And all we're really trying to get at is that your spinal cord has water on the outside of it, and it has a space running down the middle that is filled with fluid. And you're looking at basically at the cells that line that particular canal. And so when you click on this link, it'll go to the histologyguide.com spinal cord. And here, remember, you can scroll through the menu. And I have some pictures that I'll go over where I don't have to go over it on the slide. I have pictures for you. But I draw your attention to the central canal right here, because then you'll zoom right in on the cells that you're looking for. And so these are just pictures. I've made some pictures for you. So again, I'll put these up as a PDF for you guys to study from. So here's I just want to kind of take you on a little bit of a tour through the spinal cord for a second. And so I want to draw your attention to this is your vertebrae, remember. So this is your spinous process right there. And then the transverse processes out on the side. And then this over here, this is the body of your vertebrae, right? And so this is going to be inside your abdominal cavity. And then this is, all of this stuff is your back muscles. And so now we are looking at the spinal cord, which is this thing right here is your spinal cord on the inside. And then there's a little bit of fluid-filled space around it. That's the dorsal cavity where your spinal cord lives in your vertebral column. And then right in the middle, we're going to be looking at that space right in the middle, which is this space right over here. And so you're looking at these particular cells, and then they have a little bit of like cilia. There's kind of some stuff going on in there. And so those are the cells you have to identify on page 1 of your lab report, and then kind of understand the function of those cells. And you can find all of that in like the first page or 2 of your lab manual. So that's the picture that goes with this cross section of the spinal cord. And remember, you're paying attention to epithelial cells there. The next slide that you're going to look at is a smear from the spinal cord. So you're just going to be looking at some cells that were like swabbed off, like you would get off the swab of the inside of your cheek, that are literally smeared onto a slide. So we're just going to see kind of random cell bodies. That's what we're looking for in this. And then we have to identify some vocabulary, right? And so we're going to be looking for all of these vocabulary. So you're going to be looking at the cell body of the neuron, the nucleus of the neuron. And then the nucleus is also going to have an owl's eye nucleolus in it. And then there is some kind of granular dark structures called Nissl bodies. And that's all the vocabulary for this slide that you're going to be looking at. And the links take you right to a labeled picture. So here we are on the instructions again, and we're looking at the yellow spinal cord smear. We're looking at multipolar neurons, so it's going to be a neuron with a cell body that has a bunch of like sensory dendrites bringing information into the neuron, and then it's going to have one highway out, an axon, that is going to actually be sending the signal out of the neuron. And so when you click on that link for the pictured for a spinal cord smear, this is the picture that you come up with. And so you can kind of see, let me get a good color. I'll do bright green. You can see the cell body. So I'm going to use the unlabeled picture here. So you can kind of see, oops, sorry, let me get my pen active. You can kind of see the cell body here, right? That's the neuron cell body. Then you also have a bunch of little polka dotty nuclei outside of the cell. Those are the other type of cell that you find in the nervous system. And so I'm going to ask you to identify those. And then within that neuron cell body, we're going to identify a few pieces of architecture. And so there's going to be a nucleus, right? And then the arrow is pointing here at the bright nucleolus, which is inside of the nucleus. And then we have the cell body itself, which is this outlined thing in green. So this whole thing right here would be the cell body, right? And then Nissl substances are these dark kind of granular polka dots. I'll make these purple here. All these dark kind of granular polka dots in here, those are Nissl bodies, and that's basically the rough endoplasmic reticulum of the organelles in the cell. So nucleus, cell body, nucleolus. And then it's really hard to find the axon and the dendrites, and so I'm not going to ask you to identify an axon versus a dendrite. All of these things that are coming off of the cell, like this thing over here, and this thing up here, and this thing right here, and this thing over here. And then right where it says axon hillock right here, there's going to be a thing that comes off right there too. And the axon hillock is kind of like the red light, green light, the stop-go signal. And so all this information is coming in from the dendrites towards the cell body, and then it's going to exit down the axon once you have either enough signals or enough time has passed for your signals. Anyway, sensory information comes in through the dendrites and it leaves out through the black axon. And the axon hillock, it's not on your vocabulary list, but that axon hillock is really cool. That's the little stop-go signal, the red light, green light for a signal to go down your axon. So the next part of your lab is just kind of understanding some vocabulary, right? And so you're asked about the structure and function of these vocabulary words. So dendrite, axons, Nissl bodies, and synapse. So you're just kind of understanding all of that architecture. It's all in your lab manual, the first couple of pages of the chapter. And so when you think about structure, think about, is it a cell body of the neuron? Is it a process of the neuron? Where is it? Is it central? Is it outside? What is it made of? Is it a cell organelle? Is it the structure where two neurons talk to each other? Those are all structure questions, like what is it made of? And then function questions are about what does it do? So where is it sending, what direction is it sending information? Is it sending information towards the cell body where it will accumulate until it goes down an axon? Or is it sending it away from the cell body out of an axon as an action potential? So that's just kind of some basic vocabulary. Then our third slide that you're going to look at is myelinated medullated nerve fibers. And I'm just going to draw your attention to that we're going to be looking at a longitudinal section of long neuron fibers. So on some level, you're going to be having a bunch of fibers that are kind of going like this. You know, you're just going to have whatever, there's fibers, little tangle of fibers, and they're going to have some neat little structures where they kind of, the fibers come together like that. So you're looking at something cut longitudinally, like the long, like a garden hose cut long ways. And so here we are in the lab instructions. This is slide number 3 that you have to look at of 5. Longitudinal section, remember, and you can find some information about a myelinated nerve fiber in both your lab manual and also in the lecture notes, nervous system stuff. Then when you click on that link, you're going to see this picture, this myelinated nerve fiber, this myelinated nerve fiber picture. And so what you're seeing is this is a nerve fiber, right? And you're looking at a bunch of architecture. So your Schwann cell nucleus, your Schwann cell is the helper cell that helps to build up the myelin, and it's going to have this kind of little oval nucleus hanging out next to your neuron fiber. The neurolemma is kind of the plasma membrane of the cell. The myelin sheath is that protein, the protein lipoid substance that serves as insulation. And we can see some of that myelin over here. These white spaces is areas of myelin. And then there's a perfect little node. So I've drawn your attention down here to node means node of Ranvier. And this is where you can see the Schwann cell wrapping around the axon of a neuron cell body, page 253. So page 253 of your lab manual has a really nice description of what that myelin sheath gaps are. And so this area right here is an area of a myelin sheath gap. It's the little, so if you imagine your neuron axon being kind of like a bunch of little sausages, the gap where in between each little sausage, that's going to be a node of Ranvier. And that's what it looks like in the other types of cells. And it's kind of like a butterfly with a dark center like that. Does that make sense? And so you're seeing kind of the butterfly of the white myelin and then a little bit of the gap in between. That's node of Ranvier. And that is what you are identifying here. You're kind of understanding all of those bits of architecture in a longitudinal section of a nerve, peripheral nerve. So the next is just kind of understanding some of the architecture of a peripheral nerve. So it's going to ask you about what myelin's made out of. There's a specific term that the book uses. I want you to know what that is. Your lecture notes are also very helpful about all of this stuff. So take a look at those first 5 pages of your lecture notes. What is the function of the myelin sheath? What is the neurolemma? It's the plasma membrane. So what structures would you find within it? What do you find in the plasma membrane of other cells? What is the function of the neurolemma? What does a plasma membrane do for other cells? And then again, what is a node of Ranvier? Then you're going to do a little bit of neuron classification. And so you're going to kind of see in your book that you can classify neurons by structure and function. And so this is your first kind of look at the structural, the way neurons are organized structurally. And so I make a little note here that you can draw these if you want to. I'm looking at page 255 in your lab manual, and it has drawings of these. And so if drawing these shapes of neurons out helps you understand them, then that's fine with me. Do whatever you need to do here to kind of understand the difference between these 3 types. And then remember when you think about function, function is only going to be one of these three answers. It's going to be a sensory nerve, it's going to be a motor nerve, or it's going to be a mixed nerve. And so I want you to make sure you're comfortable and understand the difference between those three. The fourth slide we're going to look at is of the dorsal root ganglion. And so I have a picture this kind of jumps ahead a little bit to the spinal cord, and that's exercise 19. And so you might want to kind of glance up at page like 304. So maybe page 304 in your lab manual, and that might preview a little bit about the dorsal root ganglion for you. All right, so we're back at the instructions. We're looking at slide number 4, the dorsal root ganglion slide, and it tells you here that this structure is just outside the spinal cord on the dorsal side, on the posterior aspect of the spinal cord. Zoom in and look at the cell bodies and the nerve fibers. And so it's going to be a particular kind of neuron. Is it multipolar, unipolar, or bipolar? And it's going to have cell bodies and nerve fibers. So let's go take a look. So just a quick review. When we're talking about the dorsal root ganglion, here is our spinal cord, right? And then you're going to have some nerve fibers that come off the back aspect of the spinal cord, and then you're going to have nerve fibers that come off the front aspect of your spinal cord. Eventually, they join together, right? Out here. So we're going to have both of these colors together out there. But before that joining happens, we have an anterior root, which is the pink one, and we have a posterior or dorsal root, which is the green one back there, okay? And so what we're looking for is the dorsal root ganglion. And so that's going to be a bump. And that is going to occur right here. Do you see that little bump? This little bump right there. And it's because there is a little cell body in there. So it's like a little cell body, and then it goes down and its axons are kind of below. And so you get a whole bunch of cell bodies with axons that are going out, right? Cell bodies, axons. And so that's what I mean when over here, when I say cell bodies, I'm talking about all the little circle lumps, right? And when I say axons, I'm talking about the stringy things. Okay? That's what a dorsal root ganglion is. It's this whole bumpy critter, this whole dang thing right there. That's dorsal root ganglion. And so we're going to look at a couple different pictures of it. And so all you have to identify on dorsal root ganglion is basically be able to tell it apart from other things. That's it. You just have to know how to identify this bumpy thing with circular structures and axons in it, cell bodies and axons in it. So I just want to review for just a second on the picture we already saw of a cross-section of the spinal cord, right? And so here's your spinal cord, right? And there is kind of a butterfly inside like this, right? And so this is your gray matter, and out here is your white matter. And so all of your cell bodies are these dark polka dots that are hanging out in the gray matter. Dark polka dots equal cell bodies. And then out here in the white matter, you're going to have a bunch of myelinated axons. So if you zoomed in on this area right here, you would have a bunch of circles with little dots in the middle, a myelinated axon. So out here are axons, and in here are cell bodies. And so we're going to see that in the dorsal root ganglion too, right? So this is what the dorsal root ganglion looks like on this slide. So here you can see the spinal nerve kind of exiting, and then here we have a bunch of cell bodies right there, and then axons right there. Axons right here, cell bodies right there. So that's kind of what it, and it would go up and it would hook in to the dorsal horn of your gray matter. And we'll learn more about that next week. I just wanted to show you so that you knew what, when you looked at this slide, you knew what the dorsal horn was on this slide. And so I just gave you a picture of both of them here. So there's one flavor of dorsal root ganglion with the cell bodies and then the axons, and here cell bodies and go away. Cell bodies and axons over here. And then the very last slide you're going to look at is of a peripheral nerve in cross section. So remember, we looked at a medullated nerve in longitudinal section when we looked at nodes of Ranvier and stuff. And pardon me, let me turn my pen on. That's what this one is down here. This is a longitudinal section of a nerve. So if we zoomed in on some of the light spaces in here, we'd probably see some nodes of Ranvier in there. But what we want is the cross section. And so this one is in cross section. And so you're going to see all the layers of wrappings around a nerve. And so you're going to see a wrapper kind of around the whole outside. And then within the nerve, there's going to be some bundles. There's little fascicles within the nerve. And so there's kind of some little fascicles, right? And then on the inside, the very inside, you're going to have axons with myelin around them. So some of them are just going to be little unmyelinated axons, and other ones are going to be axons. The dark in the middle is the axon, and the white space around it is the myelin. And so we're going to see layer names for all of these layers. And it goes epi on the outside, peri around the fascicles, endo around the individual axons. So epi is outer, peri is around the fascicles, and then endo is around the axons themselves. And so here, that was the cross section of a peripheral nerve. And so when you go to this link right here, or this link right here, you're going to see the picture I just showed you. It's going to have a cross section of a peripheral nerve with its little fascicles in it, and then it's going to have a longitudinal section at the bottom. This is the one we want to look at, that cross section of the nerve. And that's where we're going to see the individual layers that wrap around the different pieces, parts of the nerve. Epi, peri, endo, remember? So I have this picture just to kind of remind you what the cross section is, right? And then the next picture goes into a little more detail. And so I zoom in on that a little bit. And so you can see epinurium out here. So this is our whole nerve. And the epinurium would be the wrapper, the wrapper kind of on the outside of all the bundles of fascicles and everything, right? And then perinurium would be kind of around the fascicles. And so there's a fascicle. You know, there's a fascicle. It's probably actually, I should probably take that off. It's probably a fascicle right there, right? And then this is a fascicle over here. And then this is a fascicle over there. And we're going to have some connective tissue wrappers in between those. And so this little tissue right in here, this little squiggly tissue, that is perinurium. This little squiggly tissue right in here, that is perinurium. That is the wrapper around the fascicle. Okay? Then endonurium, remember, is going to be around individual axons. Let me get my pen really small. So right around individual axons, we're going to see endonurium. And so this is the really zoomed in picture where we're looking at endonurium. And I'm just circling a whole bunch of axons. Some of them have myelin, like this axon right there. That doesn't have any myelin on it. but most of these have myelin, right? That is endoneurium, the wrapper around each individual axon. And, it's kind of on the outside of neurolemma and the Schwann cell and all of that stuff, but it gets kind of hazy. It's just a membrane. Just kind of understand, again, the epi is outer, the peri is around fascicles, right? And the endo is around axons. Around axons. And this is the outermost wrapper around the whole dang thing. And that's your lab for this week. So please, good studying to you. And I'll see you next week for spinal cord.